Hey students, this is Wendy Gideon. This lecture video is on inflammation. And recall, inflammation is one of the processes of the second line of defense. It's part of innate immunity. Also known as non-specific immunity. So inflammation actually has a purpose in the immune defense. I'm going to start by giving you an example of when inflammation has a positive benefit in that it's performing a very important function for your immune system. And then I also want to tell you about when inflammation goes overboard and can have a negative impact on the body as well. So starting with the overview of inflammation in a positive light, doing its job. So in this picture, we see somebody has a nail that punctures the skin. So maybe they step on a nail and that nail has bacteria on it, and those bacteria are now bypassing the first line of defense. So the skin, the anatomical barrier of the skin, is that protective first line of defense. So that's why we say inflammation is second line of defense, because something happens where the pathogen is able to get past your barriers and is now inside of the body. So these little green bacteria that you see in this picture, they are inside of the puncture wound, and these damaged cells of the skin, they start to produce chemical alarm signals. They're shown here in blue. And those chemical alarm signals initiate a chain of events. So what happens? A chain of events that leads to recruitment of white blood cells to this site of injury. And these white blood cells are able to squeeze out of the blood vessels, out of the veins or capillaries. They're able to squeeze out and migrate into the tissues, these phagocytes. And this is for the purpose of allowing the phagocytes to do their job. Their job is phagocytosis, to digest the pathogen and remove it. So these bacteria are swallowed up by these phagocytes and digested. And then the wound begins to heal as those skin cells are replaced by mitosis. So there's a YouTube video here that I like that shows this process. Let's take a look.
Leukocytes are white blood cells that help fight infection. At sites of injury, infection, or inflammation, cytokines are released and stimulate endothelial cells that line adjacent blood vessels. The endothelial cells then express surface proteins called selectins. Selectins bind to carbohydrates displayed on the membrane of the leukocytes, causing them to stick to the walls of the blood vessels. This binding interaction is of sufficiently low affinity that the leukocytes can literally roll along the vessel walls in search for points to exit the vessel. There, they adhere tightly and squeeze between endothelial cells without disrupting the vessel walls, then crawl out of the blood vessel into the adjacent connective tissue. Here, leukocyte rolling is observed directly in an anesthetized mouse. The up and down movement of the frame is due to the mouse's breathing. Two blood vessels are shown. The upper one is an artery with blood flowing from right to left. The lower one is a vein with blood flowing from left to right. Leukocytes only adhere to the surface of veins, They do not crawl out of arteries. Some leukocytes are firmly attached and are in the process of crawling through the vessel walls, whereas others have already left the vessel and are seen in the surrounding connective tissue. When the blood flow is stopped temporarily by gently clamping the vessels, we can appreciate how densely both vessels are filled with red blood cells. Red blood cells do not interact with the vessel walls and move so fast under normal flow that we cannot see them.
Okay, so that describes the process. So let's take a look. Actually, so let's take a look now at the steps of the inflammatory response. So just like we saw in the diagram, this is initiated by cell injury or cell death. So there's cell injury or cell death. And this releases inflammatory signals. And there's various types. But these inflammatory signals initiate a chain reaction of events. So usually these inflammatory signals stimulate. Now let's see if we could diagram this as well as we go. So they were talking about, here's the skin, and then there's the blood vessel underneath. And now I want you to recall a particular white blood cell that hangs out in tissues and is chock full of histamine, Do you remember a white blood cell? Let's recall. So this, in a histology stain, this particular white blood cell would stain purple and would have lots and lots of purple granules in the cytoplasm. And this is one that's in the tissues, not in the blood. Do you recall who this is? Yes, the mast cell. So mast cells would play a role in this particular example where we have a skin wound. All right, so let's follow what they were saying. So we get an injury point. So they're talking about this nail punctures the skin, and on that nail we have bacteria, and then we have all these injured cells because there's a big gash here, and so there's these inflammatory signals. So these inflammatory signals will make them blue. Okay, these inflammatory signals are released from these injured cells, and these inflammatory signals will come and they'll bind to mast cells, and that will tell the mast cell to degranulate. And recall, the mast cell degranulates. That means it's going to release these purple granules. It's going to release histamine. And histamine is another type of inflammatory signal, also known as a cytokine. So we'll say step 2 is that specific white blood cells get activated at this point. So specific white blood cells are activated, and for example, in this particular example, It's not always going to be the mast cell, okay? Just because the mast cell happens to be in the skin tissue, this works for this particular example, okay? But the mast cells underneath the skin will degranulate, and when they degranulate, they're going to release histamine. So the mast cells release the histamine, and now that initiates more of a chain reaction. So the histamine, it causes the blood cells. Okay, so remember the blood vessel is lined with all of these what are called endothelial cells. So these are endothelial cells lining this blood vessel. And the histamine interacts and binds to the endothelial cells. And when it does that, it causes the blood vessels to become leaky, we say. It actually causes the blood vessel to form little gaps in between the endothelial cells. We call that process vasodilation. So the blood vessels expand and become leaky. So fluid is now able to migrate out of the blood vessels into the tissues. And part of this as well, we get selectin proteins. Proteins form and attach to leukocytes. So in that YouTube video we just saw, they're showing you that these blood vessels have lots and lots of red blood cells that are zipping through. And the red blood cells don't have the attachment ability to grab onto a selectin. So let's draw a selectin protein attached to one of these endothelial cells. And so the selectin has the ability to grab onto one of these leukocytes. And when the leukocyte attaches to the selectin, we call that margination. Or sometimes it's referred to as leukocyte rolling, which means that the leukocyte is attached to the selectin. Let's go ahead and draw in how about a neutrophil here? So this neutrophil will attach to the selectin. There's a neutrophil. So the neutrophil attaches to the selectin, and that's what we mean by margination. So it's sticking or rolling along those selectin proteins until it finds a gap in between the endothelial cells where it can squeeze out of the tissue. And so remember, vasodilation has allowed for the blood vessel now to become leaky. And so fluid can easily leak out of the blood vessel. And that's to allow these phagocytes to leak out of the blood and get into the skin. Now, what's in the skin again? Well, I guess we should probably draw those guys in, right? It was what was attached to the nail. Remember, the nail was dirty, so the nail had bacteria on it. And those bacteria now have breached the first line of defense, the skin, and they're inside of our tissues, and they're going to start causing, they're going to start replicating and start causing damage pretty soon. So we better eat them. The neutrophils got to get there. And so the signaling cascade is allowing the neutrophils to become, to stick to the walls of the blood vessels and then to squeeze out of the walls of the blood vessels. And when we say they squeeze out, so the leukocyte rolling is sort of that sticking. So they're sticking to the selectins. And then when leukocytes actually squeeze out of the blood vessel, we call that diapedesis. Diapedesis, it means walking out. Okay, so the diapedesis process, this is when the leukocyte squeezes out of the blood vessel. And in this case, the next step would be phagocytosis, because we would want to eliminate this foreign bacteria from the wound. So specifically, this neutrophil would be engulfing the bacteria to eliminate the bacteria. So here we now see why inflammation has some very characteristic signs that you may be familiar with. And actually, let's go ahead and highlight some keywords. So first would be vasodilation, okay, margination, diapedesis, okay. Here we said histamine release. The general word for that doesn't have to be histamine, is basically cytokine. There's cytokine release. So there's some sort of inflammatory chemical that gets released from white blood cells or injured cells that triggers this chain reaction. So we get cytokine release leading to vasodilation, the expanding of the blood vessels, the dilating. That's what that means. It's dilating and allowing fluid to leak out. And then margination is the sticking, the selectin proteins forming and attaching to leukocytes. Diapedesis is when the leukocytes squeeze out of the blood vessel. And then this may also lead to usually then phagocytosis to eliminate the bacteria in the wound. Okay, so common symptoms of inflammation. Redness. So why do inflamed areas become red? Well, it's because there's increased blood flow to the area and blood is actually leaking out. So we're actually getting blood leaking out of the blood vessels, this leaky concept, vasodilation. So this is where you're going to get redness. So the skin area will become red from increased blood flow. You're also going to get swelling. It's swelling because all of this fluid is leaking out. Sometimes we call that edema. Edema, which just means fluid is building up. This fluid is coming out of the blood vessels because they are vasodilating, allowing the fluid to come out. And then with all of that, okay, with all of that excess swelling in the area, we're going to get pain because there's going to be nerve endings that are going to be in the skin, that they're going to be squeezed as a result of this excess fluid in this connective tissue, and that's going to lead to some pain in that area. And then that's also going to cause the sign of heat. So a lot of times it'll be hot to the touch, increased blood flow. That's also going to carry with it more heat as well. So think about when you get a pimple let's talk pimples. So when somebody gets a pimple, what happens? Well, you get bacteria that get deep down in your pore, and inflammation is initiated. So we get vasodilation. That pimple becomes really red and swollen and sometimes painful, and you get this swollen red pimple forming on your skin. And then maybe a couple days later, you notice it has a white head on it, and you maybe pop it because that is, let's just be honest, that's actually sort of exciting when you get a pimple that you can pop and you squeeze it, and this white pus shoots out onto the mirror, you know. And what is the white pus? Well, the pus is a product of phagocytosis. So phagocytosis is when these white blood cells are engulfing the bacteria and digesting those bacterial cells. And then, of course, these white blood cells, well, they're white, right? They're white. And so the pus that's coming out of your pimple that is white, it's because there's a whole bunch of white cells. There's a whole bunch of white blood cells that have engulfed the bacteria and are in the process of digesting that bacteria. And it forms a whole bunch of debris that you can squeeze out. Another example I like to mention is sunburns. So a lot of people think that when you get a sunburn that you're actually burning the skin or something like that. No, you're actually initiating an inflammatory response. So when you get a sunburn, many of your cells become damaged. So follow the steps of inflammatory response. Cells become damaged. They release inflammatory signals. Those skin cells release that. And then this leads to cytokine release. This leads to vasodilation. When you have a sunburn, what do you notice about your sunburn? It becomes very red. It becomes swollen. It becomes painful. It becomes hot. Notice all of that is characteristic of inflammation. So that's inflammation. Those dead cells, as a result of UV exposure, you have a whole bunch of dead cells. And the steps of the inflammatory response will play out, not to remove a pathogen in this case, but to remove these dead cells. And so these dead cells will be attracting phagocytes. And phagocytes can also remove damaged or dead cells, like in the event of a sunburn. So now you see that inflammation actually has a purpose, and the purpose being to remove either pathogens that have breached the first line of defense and now are, inside of the body somewhere, and we need to initiate phagocytosis, or to just remove any damaged cells as well. So lastly, I want to talk about when inflammation goes too far. And it's similar to fever. Actually, let's take a look at fever real quick first. So in this diagram, they're talking us through the fever response. So we have this wound. We've been talking about the wound from inflammation. Well, some of these phagocytes that are being called to the scene and are squeezing out of the blood vessels, something else they're doing is they're secreting chemical signals called interleukins. So this is IL-1. IL-1 is a type of cytokine within a family of cytokines that we call interleukins. So this is another type of cytokine, just like histamine is a cytokine. And there's a whole bunch of different types of interleukins, and they're usually numbered like this. So IL-1, IL-2, IL-3, IL-4, so forth. Don't worry too much about the numbers, but you should be familiar with this word interleukin. So it's a signal, Okay, it's a signal molecule. And it literally translates to, the word inter means between, and leukin refers to leukocyte. So it's a signal sent between leukocytes as a way that they're sort of talking to each other. But it can also communicate signals to other parts of the body as well. And that's what's happening here. It's telling us step one, interleukin 1 is secreted by the phagocytes. So in the event that we have inflammation, going back to this slide, we have inflammation, and let's say these neutrophils that squeeze out of the blood and then they digest the bacteria, let's say the bacteria have replicated extensively before this happens, and the neutrophils are overwhelmed. There's too much of the enemy and not enough of the soldiers to fight. So what happens? Well, the neutrophil is going to send out a backup signal. It's going to send out an IL signal, an interleukin signal, and it's a broadcasting signal. It goes to other phagocytes, so it might recruit the king of the macrophage, or the king of the phagocytes, the macrophage, to come and bring backup is sort of what interleukin tells the leukocytes. But it also happens to go to the brain, and it goes to the brain, so the brain is aware that there is a breach of the first line of defense. There's an infection. There's an invader. The phagocytes are working on it. However, they have sent this signal, which is basically saying there's an alarm. It's an alarm bell going off in the body of there's an invader. There's a problem. There's inflammation somewhere. There's skin cells, or other types of body cells that are being killed as a result of this pathogen, and we need help. And another layer of that second line of defense that coincides with inflammation would be the fever response. And so that interleukin goes to the brain, to the hypothalamus in the brain. And then step two, it says hypothalamus secretes some signals. Okay, so it secretes prostaglandins, okay, another type of cytokine. There's a whole bunch of different types, which resets the hypothalamic thermostat. So normal body temperature would be 37 degrees Celsius. Also, that would be 98.6 Fahrenheit. So now we're going to raise the temperature. We're going to go up. How's that happen? Well, there's a physiological process to that. It says the nerve impulses cause shivering, a higher metabolic rate, inhibition of sweating, and vasoconstriction. And then these increase body temperature to the point set by the thermostat. And so this is how the body is going to cause the temperature to increase. And remember, we talked about that in the first video, where that is part of second line of defense, sort of a generic response to an invader, because increased temperature causes protein denaturation. And so that is going to slow replication of pathogens. Proteins to denature. So that's going to slow down. It's going to slow down viral replication. In the case of a viral infection, it's going to destroy bacterial proteins. It's going to do all these things that are going to help eliminate the pathogen. Okay, now on to when inflammation goes too far. We already talked about how fever can go too far in the first video. Let's talk about inflammation going too far. So I already set up this picture earlier, so you wouldn't have to watch me painstakingly draw this out. But here we're looking at the lungs, and you may have taken an anatomy class before and be aware of what I'm trying to draw here. What I'm trying to draw is the anatomy of the lungs. So this is the trachea. And the trachea has all these cartilaginous rings. That's what I'm trying to show here. keeps this airway rigid and open, so the main airways don't collapse. And then we have the trachea that branches into the bronchi, and the bronchioles further branches into bronchi and bronchioles. So the tubes, airway tubes, just get sort of thinner and thinner as you branch out into the lungs. And then these bronchioles, these bronchial tubes, terminate in these little air sacs. And these air sacs are called alveoli. So here I've zoomed in on an alveoli, a specific, I guess it would be alveolus. Alveolus is the singular. So if we take a closer look at the alveolus, this little air sac, these are very, very fragile. They look like little collections of grapes. So there'd be a bunch of these, in here. And they are actually only 1 cell thick, so they're very, very fragile. And then lining the alveoli are the blood capillaries. So this would be a blood capillary. And then this would be an alveolus. Okay, that's what I drew here, 1 cell thick. So in our alveoli, what should be in here is pretty much just air. Okay, these are air sacs, so that's supposed to be air in there. And this is a very important part of our body, because this is where gas exchange occurs. So we breathe in oxygen, and then that oxygen diffuses into the blood and gets picked up by red blood cells. And it's also where we release our carbon dioxide waste product from Krebs cycle. So that gets released. And so we call this the spot of a gas exchange. It's really, really important. This is the only way you get the oxygen that you need for cellular respiration, for that electron transport chain to run in all of your body cells. So these blood vessels picking up the oxygen, they're gonna circulate, out to, continue the pathway all the way through the body and deposit oxygen and oxygenate your tissues is another way to say that. Well, it turns out that there's some leukocytes that hang out in the alveoli to protect the alveoli from invading pathogens. And one such leukocyte is the alveolar macrophage. So this is an alveolar macrophage. So they do hang out there, and they help with eliminating any pathogen that you might breathe in and might end up getting deep down into your alveoli. So now I want to tell you about how inflammation can be initiated here. And by the way, this is how people are dying from coronavirus. So coronavirus is a pretty big deal right now. It's having major, major impacts on the world. It's the reason why I'm talking to you in my sauna right now instead of in the classroom. So it's a really, really big deal, and people are dying. Mostly people who are elderly or are already immunocompromised, meaning that their immune system is not going to function properly. But what happens is, it turns out that the virus, the coronavirus, will make it green. So the coronavirus gets deep down into the lungs in people who die from the viral pneumonia. So this little virus, this little spiky virus here, this is coronavirus, which we're actually calling it SARS-2. So SARS-2 coronavirus, we're seeing that it invades the cells that line your alveoli. Okay, so they actually can invade and attach to these alveolar cells. And when they do that, they damage those cells so that there are cytokines that get released from the damaged cells, and those cytokines stimulate the alveolar macrophages to send out interleukins. And there's a particular signal that's showing up in the pathology reports right now. And what they're noticing is that these people, their macrophages are releasing, their alveolar macrophages in response to the viral infection are sending out a very powerful cytokine signal called interleukin 6. And interleukin 6 is going to send a message to other leukocytes like neutrophils and natural killer cells. And of course, remember what also happens after we get cytokine release. Let's go back to this picture here. So cell injury, cell death. In this case, I'm telling you that there's damage to the cells of the alveoli as a result of the coronavirus infection. Those viruses invade the specific cells of the alveoli. That is how they replicate inside there. So they destroy those cells. So the cells send out this inflammatory signal, activating the alveolar macrophages. We get more cytokine release. So then what happens? Vasodilation. Okay, so then this blood capillary is going to become leaky. And so fluid is going to start leaking out. So we're going to get fluid loss. So we're going to get fluid leaking out. And what happens to these alveoli? Well, they're filling up with fluid. Well, what's going to happen to gas exchange if you get a bunch of fluid in your alveoli? Well, there's not going to be any gas exchange. It's not going to be very efficient. What's going to happen when we unleash natural killer cells into the alveoli? What are the natural killer cells going to do? Do you remember natural killer cells release perforins? They degranulate. They release perforins and granzymes, and those toxic proteins cause cell death. So it actually causes cell death within the alveoli. And then what happens to the alveoli? Well, the alveoli collapse. So what they're seeing is that the alveoli just get completely destroyed. The consequence of this is that your alveoli is ruptured. You're also getting a whole lot of fluid in both lungs, both the right and the left lung. Let's take a look at a paper I've been reading recently describing this process. So in this paper you can see, this is from the New England Journal of Medicine, and a novel coronavirus from patients with pneumonia in China. So here they're analyzing the patients in China. This was published in February 20th, 2020. And one of the things they're doing is they're looking at patients who have the viral pneumonia infection caused by coronavirus, which is the fatal, potentially fatal symptom of the disease, the COVID-19 disease. Take a look at these x-rays. This looks crazy. So normally when you look at chest x-rays, you should see a lot of black because in the chest x-ray, because there's supposed to be a lot of air in there. And when you take an x-ray of a healthy lung, there would just be blackness. You would see the ribs, obviously, but there would be blackness within the lung tissue because the black represents just air, and you want nice, clear airways. Take a look at these COVID-19 patients with viral pneumonia. Take a look at this one, the B. Oh my goodness. Now we're seeing why these people are dying from the complication of coronavirus COVID-19, which is referred to as acute respiratory distress syndrome, because they have all this pus. There's fluid, there's pus in the lungs, and this is going to obstruct their ability to get oxygen. This is going to cause the symptom of shortness of breath, where they are literally having labored breathing, and so this is causing the fatal symptom, which is called acute respiratory distress syndrome. This is what's happening as a result of COVID-19. And then what happens is you can't get airflow. Now what can they do? Well, they can ventilate. So they can attach a ventilator that will mechanically inflate these little air balloons called the alveoli as best they can. But what's going to occur, and what we're seeing in the autopsy reports, is that death occurs from a condition we call hypoxia. Hypoxia just means a lack of oxygen. Oxygen is really, really important because this is how your cells get ATP from the electron transport chain. Oxygen is that final electron acceptor. And I thought maybe you could appreciate all of that since you have just went through the process of learning the metabolism stages and you understand the importance of oxygen. And now you're understanding inflammation. And here is where we would say inflammation has gone too far. So inflammation has a purpose. The purpose is to allow white blood cells to infiltrate an area where there's an infection and help in the process of eliminating that pathogen. But in particular, this IL-6 seems to be causing this increased inflammatory response where we're getting lots of fluid building up in the alveoli. We're seeing lots of cell death caused by the natural killer cell action. And we're seeing these ruptured alveoli. And we're seeing this effect of hypoxia, okay, where the patients are dying because they simply cannot get proper gas exchange. So there's actually a treatment that they're using right now. And I don't know if I can correctly pronounce the name of this medication, but it is an IL-6 blocker. So I was just reading about this. It's there's a medication, a treatment that they're using in collaboration with some other treatments as well, but an IL-6 blocker, so it blocks the inflammation. Here's what this medication is called. Tocilizumab. Not sure if I'm pronouncing that correctly actually, but this is what's showing up in the medical literature right now of one of their potential treatments, or a potential treatment that's working right now to block the action of IL-6, because if you block that release, let's go back to again here. So if we block the cytokine release, if we have an IL-6 blocker, it'll block cytokine release. We won't get fluid leaking out. We won't get the infiltration of some of these white blood cells that are really causing more harm than they are good, because they're destroying a very sensitive area of the body. Remember, there's only it's a 1 cell thick layer around these little air sacs. And if you get a bunch of rupturing here, you get a bunch of damaging there, and if it's significant enough, like you saw in those x-rays, yeah, that's a death sentence. So maybe that gives you a little bit of insight into the severity of COVID-19 and why there are a lot of precautions and a lot of things happening in our world right now trying to prevent more people from coming in contact with this virus. And that's all for now.